Next up, we have Maurizio from Armida Labs, someone who I reconnected with from about 5 years ago. Come on up. Great to see you again.
See you again.
Likewise.
Thank you, Matthew. I can have the first slide. So my name is Maurizio Pellecchia. I'm a professor of biomedical sciences at the School of Medicine at UC Riverside and founder of Armida Labs. Armida Labs is really based on new technology. Going back to itself, okay. It's based on new technology that is targeting a receptor tyrosine kinase for which there are really no good inhibitors so far. So as I was saying, so we are targeting the EphA2, which is a receptor tyrosine kinase from which there are no therapeutics, and is really essential for the development of cancer metastasis, which is what we are trying to address with our molecules. As the first slide will eventually show up, EphA2 is involved in protein-protein interactions and is involved in cell-cell contacts. In normal tissues, these contacts are always taking place through the ephrin ligands and the receptor, whereas in cancer cells, particularly most solid tumors, EphA2 gets overexpressed and is no longer bound to its ligand, and this is a signal for proliferation, transendothelial migration, and adhesions to other tissues, so all the hallmarks of metastasis. So it's very difficult to mimic this dimeric state that the two proteins bring on the EphA2, but we were able to do that by developing these molecules that we call Targefrin, which is, again, an ephrin mimetic that is restoring the level of EphA2. In particular, Targefrin causes degradation of EphA2, so it's a very powerful degrader. So if I go to the next slide, can I use this now? One time? Okay. So this didn't happen overnight. It's a journey of about 11 years of work through my laboratory and my academic collaborators, in particular just around the corner here at the Cedars-Sinai Medical Center, where we tested Targefrin and its evolution into the current molecule that we have patented in 2018 and 2022 that we intend to develop to the clinic. So our first indication will be pancreatic cancer, where EphA2 is really overexpressed. So of the 60+ thousand patients that will be diagnosed with pancreatic cancers, the majority will die within a year. And you see the difference on those that they don't die within a year is really the level of EphA2. A small percentage of those patients don't have EphA2. Targefrin can eliminate EphA2 very efficiently, so bringing those patients from high state EphA2 that have a very high mortality within the year to those in low EphA2. Now, so that's what gets us very excited. So we could really make a huge difference in pancreatic cancer very quickly within a couple of years from seed funding. Moreover, Targefrin has this other property that causes the internalization of the receptors. It's very similar to an antibody, but it's a micro antibody because it's still a small molecule. So we have developed strategies where we can conjugate this molecule with chemotherapy, and that's why our slogan is changing the face of chemotherapy, that will selectively deliver chemotherapy to EphA2 expressing solid tumors, aka all the metastatic solid tumors. So we have three different strategies that we want to develop. Our landscape is pretty unique. We have only one competitor that has the similar target space and a molecule that has a similar characteristics, but is not an agonistic molecule, is an antagonist. And we can discuss later the advantages of our agonistic agents, that they can cross the degradation and suppression of EphA2 levels. So these are pretty much our goals. We want to develop Targefrin first as a molecule as it is for metastatic pancreatic cancer. We have a pretty short path to a clinical proof of concept from where we are today with a relatively small investment and the seed stage. And then we could develop the targeted delivery approach using a variety of drugs. The advantage of Targefrin as a target delivery strategy is that we are not limited to monomethyl auristatin or DM1, similar to other ADCs, but we can conjugate nearly any drug. That's why we want to change the face of chemotherapy with our approach. So summary of key points. So the seed funding would really allow us to go all the way to a clinical proof of concept very quickly within a couple of years in pancreatic cancer. Most of the trials could be done here at the Cedars-Sinai Medical Center with the support of our CSO, who is actually a professor of medicine at the medical center here at UCLA and the Cedars-Sinai, and also Steve Pandol, which is our CMO. From then on, once we have a clinical proof of concept, we could then deploy the Targefrin platform against a variety of solid tumors and also the target delivery strategies from there on, perhaps in a Series A financing. These are the folks involved. Carlo is also here. He's running most of the technology in the lab. As I said, our collaborators are at the Cedars-Sinai Medical Center, and we may have got a phase 1 SBIR NCI grant to start the development of Targefrin. So I believe I was almost on time despite that. Okay. Yes.
Who'd like to start? Oh, Boris, thanks.
What's the molecular weight?
The molecular weight is of Targefrin is about 4,000, 3,000. 3,000.
3,000.
3,000. Yeah.
So does Bicycle Therapeutics have an agent against this as well?
Bicycle Therapeutics as an agent is much larger because I believe is about 4,000, but then so the agent doesn't cause degradation of EphA2. So in isolation is not active, has no activity. So they can do the first trial that we're trying to do. So their approach is to use the agents to targeted delivery of chemotherapy. So they have to add a cleavable linker and then the chemotherapy together with it. So the entire agent is about 6,000, I believe, molecular weight.
And it's a dimer like yours?
There is, it's called Bicycle because it has 2 cycles.
Yeah, but
so it's a monomeric compound, but it's much larger, yeah.
Yeah, but the 2 cycles bind one epitope, like one.
Yes, 2 cycles bind one EphA2 ligand binding domain, correct.
So what's the advantage of using the Targefrin approach versus a synthetic peptide approach?
So Targefrin is a synthetic peptidomimetic per se. And so the advantage of Targefrin is much more potent, has a higher tissue distribution, is very stable because we modified it extensively over the years, so it doesn't resemble a peptide-like anymore. And it is agonistic, that's the biggest point. Okay. So in the sense it causes degradation of EphA2, internalization and lysosomal degradation. We have done extensive in vivo imaging studies, etc. So Bicycle Therapeutics does not do that. It just simply binds there, does nothing. So you can only use that as a vehicle to concentrate chemotherapy to the tumor site. Right?
Thanks.
Which we could do as well, but we have the active internalization, so it's an advantage. So we don't rely on a cleavable linker. Right? I hope I explained it well.
Yeah, thank you.
So aside from pancreatic cancer, are there any other cancers that could benefit from this type of treatment? And I ask because the challenge with pancreatic cancer is that if it's not caught early enough, it's lethal. And I don't know if we're doing the greatest job of early stage detection of pancreatic cancer. So are there any other cancers or patient populations that could benefit from this therapy?
Yes, absolutely. I think we have preclinical data on breast cancer, lung cancer, renal carcinoma, melanoma, all published, and prostate cancer. Even the recurrence of prostate cancer really depends on EphA2. The reason why we picked pancreatic cancer as a first indication for clinical proof of concept because it's a very short duration, and the other is a group at Tulane University has developed companion diagnostics that we could use, where they use fragments of EphA2 that they enter in circulation of the patient's blood as a marker for pancreatic cancer. Because as you know, there are no markers for pancreatic cancer that are reliable right now. So we could use that as a companion diagnostic to prove that we're actually suppressing EphA2 level with a simple blood test. And the fact that, unfortunately, pancreatic cancer develops so quickly will give us a very rapid clinical proof of concept within 6 months.
Okay, and so
for prostate, etc., you need to wait many years,
right.
As you know. Yeah.
Right. And for the other cancers that you mentioned, what would be the number 2 after pancreatic cancer?
The majority of solid tumors that they become metastatic depend on EphA2 expression. All the ones we have a look at: breast, lung, renal carcinoma, pancreatic, prostate cancer, as I mentioned. Prostate cancer, that sometimes becomes hormone resistant, and that resistance develops when EphA2 is overexpressed. We have a lot of clinical data that I can share where EphA2 is really a critical player. Yeah.
So for the other cancers, you haven't prioritized any of the other ones? You just know that there could be a potential benefit?
I would love to be so forward-thinking. We have, right now, our focus is on pancreatic cancer to have this clinical proof of concept, and we hope that maybe you would agree with me that once we have that, then we can go to Series A's and go to breast will be our next indication, but we have lots of data on prostate cancer as well.
Got it. Yeah. Thank you. Mm-hmm.
Maybe I'll ask another question. You mentioned this is smaller molecule, so is it cleared from PK perspective? Is it cleared more rapidly, and do you need to do an FC conjugation like the data you're showing up there, or? Because the value proposition is you get better penetrance because it's a small molecule.
Right.
Rather than on the flip side, if you have to do an Fc fusion to get protracted PK.
In the field of ADCs, the current thoughts that you may be experienced from your question is that it's probably better to have, that's what the Bicycle Therapeutics is selling their product, to have something that is a shorter half-life, so that either you target the tumor and deliver the chemotherapy to the tumor or that molecule is eliminated. But you're right, you need to have a sufficiently long half-life so to reach the target. So we have done a number of experiments. I have a lot of data here that I can share. I believe we have it here. So the levels that we can reach of the plasma levels that we can reach on the molecules are very, very high compared to the activity of the molecule. So this is a very, very potent compound in suppressing EphA2. So on the other hand, it doesn't stay in circulation for weeks like an antibody. So it could be amenable for both eliminating EphA2 very quickly in the clinical proof of concept we like to do, but also as a drug conjugate, because it will have a sufficiently long half-life to reach the target, but then it will not stay there forever. That specifically will accumulate in some other tissues and deliver the drug in other tissues where you don't want it to be, which is the main issue of ADC. So in theory, in principle, this could be a disruptive technology in the field of ADCs, if we get there. But yes, it's pretty much what you want. So it's a sufficiently long half-life to reach the target, but not too long to create issues everywhere else. And we have done, tox studies, etc. So from a PD/PK point of view and tox, the molecule seems pretty ready for IND-enabling studies and phase 1 trials.
Yeah, go ahead. I think we have time for one more.
My question is regarding how far you want to drive this project yourself. What's your goal in terms of you want this to, you want to own this, or are you looking eventually for acquisitions? And if yes, at what stage?
So my business advisor there will probably not like my answer because I am an academic. So I do anything to see my molecules and our work to succeed, whichever way, okay, which is probably not the best business strategy. So our short-term goal is a clinical proof of concept in pancreatic cancer with seed funds. And at that point, we are open to any and each and every solution on how to bring this forward. Potentially, this could be a disruptive technology in the field of ADCs. Potentially, it can go to all solid tumors. As I say, if you ask me if I have a clear vision right now where this is going to go, I don't have it. That's why I need you.
Fair enough.
Yeah, go ahead. Excellent.
You mentioned that the journey started over a decade ago. Is the initial is the IP that's covering, and I also saw that you got an allowance on one of your patents. That's great to hear. Yeah. How old is that?
So we have a notice of allowance of a patent that was filed in 2018, and we have a new patent 2022. The other reason that we picked pancreatic cancer is because it's an orphan disease, indeed. So we will have an additional 7 years in that particular indication.
Great.
Yeah.
Excellent. Thank you, Maurizio.
Thank you.
Great job. No, you're good. Excellent.